Okay, hi. So this is going to be the revision video for D2, which is continuity and change of cells. It's an interesting topic that the three subsections don't really go well together. So it's really kind of three separate topics. So D2.1 is going to be on cell division. D2.2 is on gene expression or regulation of transcription translation. And then D2.3 is on osmosis and water potential. So they're all related to ways in which things change in a cell. So that's kind of the recurring theme. So cells change during their lifetime in terms of entering cell cycle to divide. Cells change during their lifetime in terms of expressing different genes, and cells change in terms of their kind of water movement. So I think that's where the kind of connection came in, but it does come across as a little bit obscure. Okay, so we are going to, this one is going to be crowded. So if you're trying to aim for the one-page summary, like I like to try to do, just be aware you have to write it smaller. The other two are not as dense, but there's a lot in this one. So we are going to kind of start with mitosis and actually probably spend more time on mitosis. And then for meiosis, really just look at what's unique about meiosis. So if you understand the stages really well in mitosis, it's easier to kind of go from there. So let's look at kind of starting with why. So like I said, we are going to start with mitosis. And then our first question is, why do cells go through mitosis? So what is the purpose of that? And also there's going to be a giant circle over there. So I'm writing all this kind of over to the side. So the term that you need to learn in kind of, this is more descriptive than just cell division, is cell proliferation. So the purpose of mitosis is for cell proliferation. And cell proliferation is essentially just when the rate of division is higher than the rate of death. So when we are actively gaining cell numbers. And then there's three reasons for cell proliferation. So the first one is going to be growth. So when an organism is actively growing. The second one is going to be to repair any sort of cells that have experienced damage. And then the third one is replacement. So these are not necessarily related to any sort of damage, but simply kind of a recycling cell process. So that would be things like skin cells is the best example, where we just replace them periodically. So we do go through periods of replenishing, so cell proliferating to replace that layer. Okay, so now what I want to do is, that's kind of our why. So my board's going to be about half-ish your page, under half if you can manage. So we are going to draw a fairly large cell cycle. Mine is not the world's most beautiful circle. If you have something you want to trace. If you are SL, you don't actually have to know the cell cycle, which is strange to me. You only actually need to know the stages of mitosis. So if you are SL, you can ignore the big circle, and I'm going to do the stages of mitosis down the side, and you can just take the whole space for that. But if you're HL, you do have to know about it in the context of the cell cycle. Does that fit in my video? It does. Okay, so there is my giant cell to represent the cell cycle. Cell cycle is the series of events that lead up to and include mitosis. And again, for some strange reason, this is tagged as HL only, even though it seems pretty foundational to me. But if you're SL, you only need the mitosis part. Okay, so first thing we're going to do is divide the cell cycle into two main stages. So the actual cell division stages are a pretty small piece of it. I'm not going to quite draw it to scale, only because if I drew it to scale, it would be too small to fit all the stages in. So this is M, or actual cell division, is occurring there. I'm sorry, it was probably meant to be a bit more in the center. And then all of the other stages, the whole rest of the cycle, is interphase. So all of the rest of this is the phases in between cell division. So interphase leads up to the actual division episode, and that actually consists itself of three parts. So we're going to have G1, S, and then G2. So G1, G2. Then the actual stages of cell division consist of mitosis and then cytokinesis. So we have 4 stages of mitosis. 1, 2, 3, 4, plus cytokinesis. So that's the cell division component of it. Okay, so we're going to go through the cell cycle, and as we go through the cell cycle, we're going to do regulation of it as we go. And again, for the next kind of 15, 10, I don't know how long this is going to take, everything related to this circle is going to be HL until we get to mitosis. So fast forward until I start writing in those lines, and then pick back up again. Okay, so in G1, so if the cell is going to start to work towards division and we want to not end up with just a bunch of tiny cells, the cell is going to grow during this time. So G1 is going to be cell growth. And included in that time period is going to be that we're going to make building blocks. So we're going to increase the number of proteins and increase the number of nucleic acids. All right, nucleotides, maybe not I'm going to be sure to write nucleotides because we don't actually turn them all the way into DNA till the next stage. And then we're also going to increase organelles, in particular mitochondria. So it's going to need to increase the mitochondria to progress through the cycle. If it's a plant cell, it's also going to make more chloroplasts then. Okay, so as it's going through that, when the cell is entering the cell cycle, it is going to periodically have to stop and check that it is fit to continue. So somewhere around here, there's going to be a stopping point. And I do recognize I'm probably going slightly off. That's all right, I can go up to there. So there's going to be a stop to check that it's actually meeting the requirements. So this is going to be a checkpoint. So it's going to stop and check, does it have the organelles and the building blocks that it needs to progress? Is this cell supposed to go on? If it fails that checkpoint, then it will exit the cell cycle and it'll go off instead into what's called G0. So G0 just means that cell just needs to go back to being a cell. So that cell is not needing to replicate. So it's going to leave the cell cycle and just go back to normal cell functioning. So this is going to be cell cycle arrest. And remember, most of our cells are in G0. Because when they're in G0, they're doing their normal job. So your brain and heart cells, once you're fully grown, stay in G0 because it's really important that they do their job. So G0 is not a bad thing, it's a good thing. If they do pass the checkpoint, what that's going to do is that's going to actually allow it to progress. And the way it allows it to progress is by activating something called CDK. So we're going to have a look, really wishing I had made this a little bit less wide so that I could have a little bit more room for my regulation here on the side. Okay, so if it meets the checkpoint, what it actually does is it actually starts producing cyclin. So when the checkpoint is made, it will start actually producing cyclin, which is a messenger, a little protein that says we have met the requirements. So that's my cyclin. So in the event that the checkpoint is successful, it will actually start producing this cyclin. Cyclin is important because it is what is needed to activate this protein called cyclin-dependent kinase. So this is an enzyme, because it ends in ACE, called CDK. And under normal circumstances, it is inactive. So this is my inactive CDK. And so cyclin-dependent kinase activity is needed to progress to the next stage of the cell cycle. So under normal circumstances, if the cell has not grown enough and isn't ready, the CDK is inactive. But once that cyclin has been made, it is going to come over here and it's going to bind to it, and that is going to activate it. So when it has cyclin attached to it, that actually wakes up the CDK. So now he's happy and alert. Woo! So ready. And when CDK is active, it will progress the cell cycle. And so when it is in that active phase, it will say, all right, let's start the S phase. So that is how that is actually regulated. That's how we regulate the cell cycle. So I've just shown one, but there's actually different CDKs and cyclin-dependent kinases at each stage. And so this would be like G1 cyclin, and it would be cyclin-dependent kinase A or 1 that is activated. You do not have to know the names of them. You're welcome to look it up. There's essentially 4 main cyclins that interact with different CDKs. Some call them by numbers, some call them by letters, so it quickly gets confusing, so you don't need to know that. Just that there are different cyclins and CDKs regulating each stage of the cycle. So this little example here has shown that as the cell reached the checkpoint and was in fact big enough that it was able to progress through the cell cycle by activating the CDK to start the S phase. So that is what would happen there. If it fails the checkpoint, G0. Okay, so let's say it passes the checkpoint, hooray. It is now going to enter the S phase, and our S phase is going to be DNA replication. So again, up here, we already made the nucleotides, so now it can replicate it. So when we say DNA replication, basically what that means is the making of the sister chromatids. So when we go into the mitosis part, you are going to need colors to represent different chromosomes. So pick your 4 because we're going to use them all throughout. So I have 4 strains. We're going to show 4. In your body, of course, it would be 46. So these are my 4 strains of DNA. So one of each of the separate strains. So when we get to meiosis, we would recognize that those 2 might actually be like homologous chromosomes. But remember, in mitosis, homologous chromosomes don't matter. They never interact with one another. So it doesn't really matter. These are just 46 separate chromosomes. And then during the process of DNA replication, each of them is going to get copied. So we are going to copy each of these. So this one would look like that. And then they will literally get attached to one another, like so. Like that. So that is what's happening during DNA replication. The actual process of DNA replication is in D1. So if you hear that and you're like, oh, that sounds interesting, tune into D1. But right now you only need to know where it fits into the cell cycle. Okay, so then each of these now are called sister chromatids. And that term is going to be super important as we go through mitosis and meiosis. So those are my sister chromatids there. And so each of these is a set of sister chromatids. And then we'll go ahead and also label that the proteins holding them together is the centromere. And on each side of the centromere, you have further proteins that is what the actual microtubules are going to attach to, called the kinetochore. We'll just go ahead and label it here because my mitosis diagrams are going to be even tinier. So that's kind of what happens. So we have made it, and then they come together, and we have made the sister chromatids. The other thing I'm going to draw on them because it's going to be important when we get to our actual mitosis diagram is we're going to show that they are held together by something called cohesin. So they have these little proteins that hold them together. And that's how they're going to stay together until it's time for them to separate. So that's cohesin, like cohesion, but without the O. So cohesin is holding them together. So they formed, they joined together, they're clustered by proteins at a centromere. There's extra proteins on the kinetochores, which are on the ends of the centromeres, to get them ready to attach microtubules. And then there's cohesin holding them together. Okay, so I think, I'm always checking that I covered everything. The only thing to note is that this is all happening in the nucleus and that they are actually still as loose chromatin. So I've tried to draw it like not too tight, but I wanted you to be able to see it. But in reality, they would all be inside the nucleus, and they would all be kind of on top of one another. So I've drawn them kind of distinct just to make a point for now, but do recognize that in reality, it would not be separate. It would be like, woo, there's this one, and then sitting on top of this one would be this one, and then on top of that one would be this one. So we're just seeing that in reality, they're not visible and separate at this stage. They're kind of joined together like that. Okay, so that's still going on in the nucleus, and they are still in chromatin form, which is the unraveled form that they are at that point. All right, I think that's all things we need to know about DNA replication. There is going to be a couple more checkpoints along the way. So somewhere along here, I'm just going to make it here, there is going to be a stop and a checkpoint that's primarily going to check for if there's any damage to the DNA. So as it's getting ready to replicate, it's probably earlier than that, I just ran out of space, but you don't need to know the exact locations. There is going to be a check to make sure that this is good DNA and to repair any damage. And then there's going to be another checkpoint kind of towards the end as well that is going to make sure there was no errors in the process of DNA replication. So there's going to be a couple more checks, and then there's going to be, of course, new cyclins produced as well. So as this is happening and as this is done, it is going to be producing the next kind of cyclins. So it's going to be like, ooh, here's some cyclin, here's some cyclin, here's some cyclin. So when it's passing, the cyclin that's being made, of course, is going to activate the cyclin-dependent kinase, and that's going to move it to the next stage. So same kind of idea that is going to cause the progression. So we're just showing that again to emphasize that that happens kind of at the end of each stage. So it's happening to move us into the S phase, and then it's happening to move us into G2. You don't really need to know anything about G2, which is great. It is kind of more growth and more preparation. So more organelles are made, so this would be where there's kind of further extension of organelles, and there's some things happening with the centrosomes. So the centrioles are kind of joining together and starting to kind of cluster and become visible. So just kind of as long as you know grow and prepare, you don't need to worry too much about G2. Know it's there, but really need to know G1 and S pretty well. Okay, so now we are going to everything that SL needs to know. So that, SLs you need to know about DNA replication, that is a necessity, but you don't need to worry about G1 or G2. Okay, so let's get to now M for mitosis. So the first 4 stages are true stages of mitosis. So we're going to have prophase, metaphase, anaphase, and then telophase. So those are the 3 stages that are, the 4 stages that are mitosis. So mitosis is specifically the nuclear division. So that is the division of the nucleus is mitosis. The last stage is division of the cytoplasm. And that is technically, I'm going to have to write it this way because I can't fit at that angle. That is technically not a stage of mitosis, which is cytokinesis, but you do have to know about cytokinesis. So we will cover what happens there as well, but it is worth noting that it is not a true stage of mitosis. Okay, so what I want to do now is use over to the side, draw the actual stages of mitosis. So I'm probably going to erase that just so that I don't have to draw tiny, but just try to end at about halfway of your page. So you can go past the circle, that's fine. I went all the way down here, and then I'm going to go across for cytokinesis. So you can go a little bit further down, but just don't take up the whole page. But I'm going to try to keep that there and just give myself a little bit more room so that I'm not having to write so tiny. Okay, so we are going to now look at the 4 stages of mitosis. Everyone needs to know these, and you need to know them at a somewhat intense level of detail. So we're going to try to condense it as much as possible. So again, I'm going to try to draw them and label. I'm not going to write separately. The labels are going to be all the explanation that you need. I'm probably going to try to draw the circles ahead of time, which you might want to do the same, just so that I monitor my spacing. So I'm going to say prophase, metaphase, anaphase. I might have to do telophase and cytokinesis over here. I think I fit them in together over there. And then we'll go to cytokinesis. Okay, so again, if you want to try to go around, but I just am running a little bit out of space. Okay, so stage 1 is prophase. And in my class, we do a cheer. So we say, please make another touchdown. So please is all in the center still. So please is saying there's things going on to prepare. So as we go, please is for prepare. So prophase is prepare. So we're just preparing some things that are going to need to happen for division. So there's 3 things you need to know about prophase. So first of all, the nucleus is constraining the DNA. So if I need to spread it out, I want to unpack it out of the nucleus. So it's like putting all your homework in your backpack, but if you get home and you actually want to do your homework, probably need to take it out. So we need to get it out of the nucleus so it has more space. So we're going to show that by drawing the nuclear membrane dotted. So we're going to label that nuclear membrane dissolves. And there was more information about how that happens in A2, I believe, that it basically wraps up into little vesicles and goes off in little vesicle time and then returns in the same little vesicles. So just a little bit of information if you were wanting to know how that happens. Next thing is that we need to get these centrosomes together. So centrosomes are at the centrioles in an area that starts to produce microtubules. So sometimes they're also called like microtubule assembling centers. I prefer centrosome. So we're gonna call them centrosomes. So centrosomes assemble and start making microtubules. So if you were to see the word instead microtubule assembling centers, that's where that comes from. So I'm not going to use that all throughout, but if you saw that word, centrosomes is perfectly adequate. They mean the same thing. So the centrosomes are assembling and then they're starting to make little microtubules, like so. Okay, and then the third thing that you need to know is that my DNA is going to condense and become visible now. So whatever your 4 colors, if you want to keep going with those. So instead of looking, even have drawn them there, they should look like nice and thick little, like this. Like that. So we are going to see my DNA is going to condense, which means it's going to wrap tighter around the histones. So I don't really know why I've drawn them like H's instead of X's. More like that. Okay, so those are going to assemble or condense. So that's kind of my third event that is going to happen, is going to be the chromosomes condense and become visible. So that's going to be important if you are ever looking at a microscope. The way you can tell it's now in prophase is by the visibility. So this is what you would be looking for under a microscope. I'm just going to write microscope to remind you that if you're given an image and you're like, how do I know if it's in prophase or interphase? If you can see visible lines, you are looking at prophase. So chromosomes condense and are visible. The way they condense is by wrapping around your histones. So I'm just going to make a little note of they do that, and that's something we covered earlier in A1, is we talked a little bit about DNA condensation. So the histone proteins, it loops tighter around those, and then they kind of tighten up as well, and that's how it gets into that kind of condensed state. Okay, so those are your three things that happen in prophase. From prophase, we go into metaphase. And metaphase is the one where they kind of have a slightly more detailed view on metaphase and anaphase of what they want you to know. So they're going to end up in the center. So it's quite hard to draw it because we're just going to kind of draw what happened in the end. But my analogy is basically we have the sister chromatids. Someone grabs this end, someone grabs this end, and then they try to move to opposite poles until that person ends up in the center. It's kind of hard to show. So it's basically like they're getting pulled from this end on this one, this end on this one, and then that ends up lining them up in between because at least initially, the actual centrosome, the centromere is stronger than the pull. So as the microtubules move, it actually ends up orienting these in the center. So let's draw them in the center. So now here's my chromosomes. And again, it's going to be hard to go into like amazing detail about that without this video going for like 9 million hours. So we're just going to make a note of that. You can kind of go back and look over that concept. So there they are in the center. So what brought them to the center is the microtubules or spindle fibers. So the IB is using the word microtubules instead of spindle fibers, saying basically the microtubule is the name of the protein, the spindle fibers like what it creates. It's half of one, six of another, half a dozen of one, six of the other. I am drawing a few extra microtubules, and I'm going to draw some little motor proteins because that is actually what is like pulling them the different directions. So I'm not going to go into tons of details, but I am just kind of reminding us that those exist so that we know that. Okay, so the two events I want you to know here well, they're kind of one event, but basically related to the same thing. So I want to say that the chromosomes are pulled to the center, and they're pulled by the microtubules. So as the microtubules move out, that ends up aligning them. And really, honestly, if that's a bit confusing, just memorize that. So end up in the center, why microtubules? And in terms of what actually allows that to happen, the microtubules are moved, motor proteins move the microtubules. So it's a little dot point in the syllabus that you do need to know that the movement is due to microtubules, and you do need to know that the motor proteins are what do that. You don't need to know a lot of details about how, but basically there's little proteins in between the spindle fibers that attach to the chromosomes, and they actually are what are pulling it this way. So they're pulling it towards the poles, which is important for the next stage as well. So right now, they're lined up in the center, and right now, at the point where they are at here, so I'm looking for my right color, they're staying together because remember there's cohesin between them. So I'm going to draw the little cohesin. So they're lined up right in the center now, and there's cohesin that's holding them together. Metaphase, once they get to the center, is going to be a pretty short stage because this doesn't stop at the center. They keep trying to pull further, and eventually the pulling of the centrosomes pulls enough that it breaks the cohesin, and then they split and the actual cohesin breaks and the sister chromatids are separated. So that is what is going to happen in anaphase. Okay, so there we are in anaphase. So now it's pushed a little bit too far out. That is the point. But the pushing of it that far out has actually been sufficient to snap the cohesin. So they are actually separated because of that. So the pressure of the migration snaps the cohesin, and my sister chromatids are broken apart, so the centromere no longer stays keeping them together. So that's event number 1, is that we snap the cohesin. So I'm going to show, well, first of all, let's make sure we connect these to their microtubules, like so. And then I'm going to show here in the middle that my cohesin broke. So there's some broken cohesin, just to show that it's no longer holding them together. So that's the first thing that happens, is that basically further migration of the tubules breaks the cohesin. So now they are broken apart, and that is what actually separates the sister chromatids. So separating sister chromatids. Okay. The next thing is that once they've separated, we then want to pull them away, further away from one another. But if you just kept expanding out to do that, the cell would get too elongated and it would pop the cell membrane. So once they are separated, they are then pulled to the sides by shortening the microtubules. So shortening microtubules, I'm trying to see if this is going to fit. Barely. Shortening microtubules pull the chromatids to the poles. So it doesn't look like you need to, it is actually the kinetochore that does the shortening. So the kinetochore protein that's still there on that side, once they've broken, it starts chopping away the little tubulins of the microtubules. But if I keep going into this video is gonna go all night long. So that's probably all you need to know, is that they pull apart because the microtubules get shorter once the split has happened. So a little bit of fussy detail there in the metaphase, anaphase area for you to kind of wrap your head around. After that, it gets a little bit easier, which is nice. So then we're gonna go from that, and I probably have a little bit more room than this. Okay, so then from there, the last stage is going to be telophase. Telophase, thankfully, is fairly unexciting, so we don't have to write anything earth-shattering about telophase, thank goodness. Okay, so there's telophase, and essentially 2 things happen that you need to know about. Now that we have actually separated the DNA, we can actually go ahead and reform the nuclear membranes. So nuclear membranes reform. Like so. And then the second thing that happens is the DNA is going to go ahead and relax, because when the DNA is all tightly coiled, it is not able to actually use the DNA to make any proteins. So it's going to relax back out, like so. And so instead of looking all tight and condensed, it's going to be relaxed back to chromatin. So the DNA relaxes back to being chromatin. And now we have successfully divided the nucleus. The last thing that we need to do is just divide the actual cytoplasm. So from telophase, it then enters cytokinesis. Cytokinesis is not actually a phase of mitosis. So it is a phase of cell division, but not a stage of mitosis. So mitosis is only the nuclear division. It's a really small nitpicky point, but you just do want to know it, because if you start talking about cytokinesis in a prompt on mitosis, you won't get any points for that. So now we enter into cytokinesis, and we are going to actually divide the cytoplasm. You do need to know that it is different whether it's an animal cell or a plant cell. So there's my animal cell. Here's my plant cell. Okay, so the way I've drawn it, the other thing I want to add, cheating. The other thing I want to add is that it is a usually even division of the cytoplasm, but not always. So it's usually an even division of the cytoplasm. But when we say usually, that just implies there are exceptions. So the two exceptions that are in the course outline that they could ask you about is human eggs. So when we're forming eggs, most species, in fact, not just humans, try to make a really large egg. So in order to do that, the egg is actually going to take all the cytoplasm making polar bodies. And then the other possible exception is yeast, which does budding. So those are the two that you could know about for uneven division. Okay, so then we have our cytokinesis right there. So we are going to then look at what the difference is. So again, this is an animal cell. It uses muscle fibers, so that's actually actin and myosin. So those are going to be some muscle fibers, and the muscle fibers are then going to create a cleavage furrow. So they're going to use some muscle to basically pull that in and create that cleavage furrow, and then it's going to split. In the plant cells, they have to create a new cell wall. So this is going to be my plant cell. And in order to do a new cell wall, they're going to line up all these vesicles. So vesicles are going to be used, and then the vesicles are going to become a cell plate. And that cell plate will end up replacing or forming the cell wall that's going to be needed in the inside. Okay, so I'm going to erase because I'm out of room, but hopefully you're at about the halfway of your page. So then we are going to summarize what we made. I just don't have any room right here, so we're going to summarize it, then compare it to meiosis, and then we'll talk a little bit about what happens if it goes wrong for both. Okay, board erase. So in order to kind of compare between mitosis and meiosis, I want to really focus on the results. So we just did all of mitosis. So let's quickly summarize what the result at the end of that process. So at the end of mitosis, we have formed two genetically identical diploid. That means they still have the full set of DNA. So they still have the homologous chromosomes, so full set of DNA. So they still have one copy from mom, one copy from dad for every single different chromosome. And they are body cells, and our fancy word for that is going to be somatic cells. Okay, so now let's use that to compare it to meiosis. So again, a lot of things are similar. So for the meiosis part, I'm not going to redraw all the stages. We're going to just really focus on, okay, we understand mitosis, what's different in meiosis. So we're going to say, but in meiosis, okay, so in meiosis, instead of having 2 cells, we are going to have 4 cells. Instead of having genetically identical, we are going to have genetically unique cells. Instead of them being diploid, they are now going to be haploid. So they are going to have only one of each chromosome. And instead of them being somatic cells, they are now going to be gametes. So all 4 of those things are different if we're talking meiosis. So the outcome of meiosis is going to be completely different. So then let's look at how. How do we create that difference? So how do we change meiosis to make it different from mitosis? So how do we get 4 cells? So the way we get 4 cells is 2 rounds of division. . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . So instead of doing one round, we're going to do two, and it literally goes through those same stages again. So it's going to go prophase 1, metaphase 1, anaphase 1, telophase 1, and then it's going to go prophase 2, metaphase 2, and dot, dot, dot. So it's going to go through PMAT twice to get there. What makes it unique? So the uniqueness of the gametes is due to two processes in particular. So they are going to be different from each other because they don't get the full number of chromosomes, but we're also going to go ahead and attribute largely this is due to crossing over, which we're going to talk about again in a second. And also it's going to be different between each round because of random orientation. So within one round of meiosis, the fact that the 4 are different is really more due to crossing over. Between rounds of meiosis, it's really due to random orientation. So it's mainly crossing over, but we'll make a note plus random orientation, and we'll talk more about that in a minute. Then how do we end up with haploid cells? So how do we end up with half the amount of DNA? So we already kind of covered that by saying 2 rounds of division, but there's going to be 1 round of replication and then 2 rounds of division. So we only replicate the DNA at the beginning and then do 2 rounds of back-to-back division without replication again. There is kind of a vocab term you'll see for this, for the fact that it does end up creating cells with half the genetic material, which is that it is a reduction division. So it is a division that does reduce the genetic material. And then we don't really need a how for gametes. Let's just make a note here that these are going to be, it's more of the why. These are going to be used for sexual reproduction so that the actual zygote ends up with the right amount of genetic material. Okay, so basically it's very similar. So what we're going to do is just clarify the difference between meiosis 1 and meiosis 2, and then we're just going to draw the 2 stages that are most distinct, which is the beginning of meiosis 1. After you get through that, meiosis 2 is basically mitosis. So we're just going to separate sister chromatids, so it's going to look the same. So we're just going to say what's the difference, and then just draw 2 stages that really highlight the difference. If you want more, go back to your notes, but again, we're just trying to kind of make this somewhat of a summary and not an endless recovering of everything. Okay, so when we get to this, we're going to make a note that it is similar to mitosis, but we are going to clarify that there are the 2 rounds. But meiosis 1, and this is really the difference, because in mitosis, all we separate is sister chromatids, but meiosis 1 is actually going to be the gathering and separating of homologous chromosomes. So meiosis 1 separates homologous chromosomes. And so when we did mitosis, I never even wrote the word homologous chromosomes up there, because they come into no contact with one another. So that's not really a part of mitosis, but it's going to be a big thing here. Meiosis 2 is going to be like mitosis. Meiosis 2 is then going to separate the sister chromatids. So it has the 2 stages, and they are different in what they separate. Okay, and then like I said, because this is so similar to my mitosis, we're not going to focus too much on that, so we're just going to draw 2 stages and leave some room at the bottom. It doesn't need a lot, but don't use up all of your room. I'm probably going to erase just so I don't have to write tiny, but try to leave some room. Okay, so the 2 stages of meiosis that are unique are going to happen in meiosis 1, and that's going to be prophase 1 and metaphase 1. So we're going to show those 2 stages because they are the unique stages. So prophase 1 and metaphase 1. Okay, so again, how are they unique? So in prophase 1, they're going to be all the other things that were going on before are still happening. So we're always going to remember mitosis. So we're still going to be dissolving our nuclear membrane. Our centrosomes are still going to be forming, etc. But the other thing, sorry, there should probably be another one for accuracy's sake. There's another centrosome. All right, and then the other thing is that this time the homologous chromosomes are going to pair up. So previously they were unrelated to one another, but now they are actually going to join together. So they're going to join together and they're going to basically kind of fall on top of one another. Sorry, this should be here. So they're going to come together and kind of literally overlap with one another. That's going to be how crossing over can occur. So that's going to happen with every pair of homologous chromosomes, is they're going to have this occur. Okay, so let's label a couple things there. So that's kind of the key event. This actual joining together of homologous chromosomes is called synapsis. So synapsis is going to be the pairing of homologous chromosomes. When they are paired like this, we now call this a bivalent or a tetrad. I think IB textbooks are using bivalent. So that's just when they are joined together. So when they have come together, they are called a bivalent. The reason why this is important is because this is where crossing over is going to happen. So this point here, where they actually overlap, is called the chiasma. And at the chiasma, that is when they are going to exchange genetic material. So it's going to be the exchange of genes between the homologous chromosomes. So the two homologous chromosomes are going to swap DNA, and that is crossing over. And crossing over is really important because if it were not for crossing over, then two of the gametes would be identical every round. So by having crossing over, we're going to ensure all four are different. Crossing over also happens at a different place every round, so it creates a lot of variation in our offspring. So that's what you need to know about prophase 1, is that the homologous chromosomes come together and that then the crossing over occurs there. Okay, in metaphase 1, two things. So I'm actually going to show an either or. So we're going to show that they line up in pairs, and we're going to show that the order can vary. So it could line up like this. And mind you, crossing over has already occurred. So I'm just being, if you don't want to draw that, you don't have to, but I'm just trying to make sure I don't leave any inaccuracies. So they could line up like this. In which case that's going to give one set of combinations, because we're going to end up with gametes that have kind of pink and blue combos or gametes that have orange and green combos. But the next time that meiosis happened, they could flip. So the next time meiosis happened, I'm still going to draw these two up top, but this time it could be that the green lands on this side, which by green or blue, we mean like mom's version instead of dad's version lands over here. I don't know why I drew that the wrong color. Sorry, my bad. Like so. So that is just showing that those are equally likely events. Of course, if you were drawing this or writing about it on the test, all of the same things are happening. So this is happening because of motor proteins, blah, 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 blah. That's why we went into the detail about it in mitosis. So you know it's the same kind of processes that allow that to happen. The key thing here is this idea that it could be either arrangement, and that is called random orientation. So by having two different possible combinations for only the two chromosomes, if you were looking at across 23 pairs, you have an almost infinite number of different combinations. So this is going to increase combinations, and that is going to be a source of genetic variation. And genetic variation is going to trigger natural selection if that happened to be a positive or helpful combination. Okay, so that was pretty brief on meiosis, but I think you have the main things, which is what are the differences in what is made, and then these two events that are really important. The bottom of the page, we're going to look at what happens if something goes wrong. So I squished this in a pretty small amount of space, so far we've been drawing everything like it's all going smooth sailing. But now we're going to say what happens if there's an error occurs during cell division. So what happens if something goes wrong? These are not the only two way things can go wrong. These are just the two things you have to know. So we're going to cover them kind of together. So one of them is something that's going to go wrong in mitosis. One is in meiosis. Interestingly, if you are SL, you don't need to know about cancer and the stuff that goes wrong in mitosis, so you can just do the meiosis, but you do need to know about the meiosis. So a bit random. So the two different kind of problems that can occur. So if the problem goes wrong in mitosis, so if we're going along in mitosis and there's a problem, the problem that you need to know about is uncontrolled division. So uncontrolled, too much. We use the word uncontrolled rather than just too much, because the key is it could also be like faulty cells being divided. So not having the regulations to know not to do these things. So the end result of that is going to be cancer. If the problem happens in meiosis, generally isn't going to form cancer, is going to instead form kind of faulty gametes. So in meiosis, the problem we're looking at is not too much division, but a problem with the dividing. So failure to separate. So things do not separate correctly. So there's a failure to separate either the homologous chromosomes or the sister chromatids. So some sort of incorrect kind of separation, and the result of that is non-disjunction. I'm just making sure I'm still visible here. So those are kind of our two errors. Again, I'm going to make a note over here that this is HL only. So you can just tune out for a second. Well, that's very interesting information. Okay, so like everything in biology, it seems like, when I say what does it start with, it starts with a mutation. So all problems, or natural selection, starts with mutation. So there's going to be a mutation occur. And there's two mutations that are particularly prone to causing problems. So mutation 1 is that the mutation is in a proto-oncogene. So this is a gene that is responsible for encouraging cell division. And so if it happens in a proto-oncogene, we now call it an oncogene, because that's fun and confusing. And that's going to cause too much acceleration. So now it's going to go through the cell cycle too enthusiastically. The alternative is that it happens in a tumor suppressor gene. So this is a gene that is supposed to catch errors and stop division. And if a mutation happens in that, then it loses the ability to stop it. So this is going to be no brakes. So we're just using driving analogies. So if you had a car that every time you put your foot on the gas, it went shoo, that would be problematic. You would run into things. If you had a car that when you put your foot on the brake, it did nothing, you would run into things. So two bad outcomes. So both of those are going to lead to problems. So as a result of either of those two issues, what is going to happen is going to be the formation of a tumor. So if these happen, So either of these, what's going to happen is there's going to be a primary tumor. So a cluster of cells that are over-replicating that have no function. So primary tumor, loss of function, cluster of cells. At this stage, when it is a primary tumor, it is benign. So that means it is not actually spreading. So when it is benign, it is contained or not spreading. At some point, though, that tumor sits there long enough, it's at high risk of spreading, and that process of it spreading is called metastasis or metastasize, depending on whether it's a noun or a verb. You do need to know that term. So metastasis is the spreading via the bloodstream. So the tumor gains access to the blood, it vascularizes, and then metastasis occurs. And then as a result of the metastasis, it travels to a new location in the blood, and it forms a secondary tumor. And that secondary tumor is now malignant, which is what we tend to think of as cancerous, so requiring more intensive therapies. So you do need to know the terms benign, malignant, and metastasis. So lots of little vocab there in terms of the cancer terminology. Okay, so then what about a non-disjunction? So non-disjunction is basically a spindle fiber error. So here we are at non-disjunction. If we have two copies of chromosome 21, for instance, so this is chromosome 21, that's the sister chromatids, and then we also have dad's copies of chromosome 21, and what's happening here is that there's some sort of failure to divide. So there is no spindle fiber, and so as a result, they are going to both move into one side. They may then divide during the next phase of mitosis or meiosis, but they don't there. So the result of that is going to end up being, in the long run, you're going to have gametes with 2 21s in this case, and then if fertilized, the zygote would have 3 21s. Sorry, I didn't actually write the word 2. And so if you end up with a zygote that has the wrong number of chromosomes, that is where the problems arise. And if it was 21, that would be Down syndrome, which is the one they said you would need to know as kind of your example one. But do bear in mind this happens with all the chromosomes. It's just normally non-compatible with life and is the cause of infertility or miscarriage instead of kind of chromosomal abnormalities. The other thing we will note is that this does increase in frequency with maternal age. To some degree with paternal age, but not till they are much older. So it's a more prominent issue there. Okay, that is all things D1. The other two will be a little bit shorter, but that was a lot to put in there. So I'm going to erase and then be back with D2, D2.2. Okay, so moving on to D2.2. So D2.2 is HL only. So if you're SL, fast forward a little bit. So that's nice. Nothing for this for you. But for HL, you do need to know all of this. It's not quite as dense as the last one. So we're going to start with a few definitions, and then we're going to basically look at kind of 3 levels of regulation. So we'll define everything, and then we'll look at kind of how gene expression is regulated, both it changes to the DNA, then it changes to whether or not you transcribe, then at whether or not you translate. So kind of 3 levels of that. So let's start by just making sure we remember what gene expression is. So gene expression is the using of a gene to make a protein. So just reminding that what that means. So it's using a gene to produce a protein. So it is the same thing as protein synthesis. It's just a different kind of phrasing for it. So while we're on terminology, I want to go through one other kind of set of words that you need to know. And again, this is D2, so it's continuity and change of cells. So every cell has a genome. So a genome is all of the genes that it has. So that's going to be all of the different genes that it has. So genome relates to the genes. Then from those genes, the cell is then going to have a set of mRNAs that it can make. So that's going to be a transcriptome. And in general, a transcriptome is bigger than the genome because of things like alternate splicing. So these are mRNAs. So essentially one gene might be able to make multiple different mRNAs depending on the splicing. So the possible transcripts that it can make is always going to be higher than the genes. Similarly, it's then going to have the set of proteins that it makes, and that's going to be its proteome. So the proteome is going to be all the possible proteins. And again, that is going to be greater because of things like protein modifications and so forth. So one mRNA could make multiple different proteins. This is where the expression part comes in. So the actual expression of which ones that are actually made, so it has the ability to make all of these, but this process of regulating this process is what leads to cell specialization. So if every cell made all of the proteins it could, so if every cell used everything in its genome and just produced all the different mRNAs, then made all the different proteins, it would be really busy, and that would be an unspecialized cell. So the choice of which to use is what actually gives you a phenotype. So the phenotype is the choice involved in that. So basically, which genes become which mRNAs become which proteins equals the phenotype. So the actual expression of the genes equals the phenotype. So understanding that the genome doesn't drive the phenotype, it's the expression that drives the phenotype. So which genes are expressed is what determines what a cell looks like. So a little bit confusing. We'll go through that one more time because I want to kind of pick that up again. So where I said the regulation, that's kind of the next big term we need to look at, which is gene regulation. So gene regulation, so we know from up here that the cell has all these potential genes, all these potential mRNAs, all these potential proteins. So the question becomes, do we express it or not? So gene regulation is to express or not to express. So it's anything that is in place that has some impact on what proteins end up being made. So it's a process that regulates that. And so based on this, so essentially the outcome of this, so as it decides, is it going to express or not? So the particular pattern that results, so as a result of gene regulation, you will have a specific pattern of expression, and that specific pattern of expression is called epigenetics or epigenesis. So if we come back, I use analogies a lot that a gene is like a recipe. So this would be saying your genome is essentially all the different recipes that you have. The transcriptome would be all the recipes that you could copy out and make slight changes to as you go by omitting certain things. Proteome is how you could use those different recipes to make different proteins or different dishes that might vary a bit if you change certain things kind of in how you cut it up. It could look a little bit different. But at the end of the day, you cannot go in every day to your kitchen and decide you're going to make every recipe you have. It's not possible. So as you go through the cookbook and you say, which recipes work for me and my family and are things I enjoy eating, those are the ones I'm going to keep, those are the ones I'm going to make. You build your actual kind of kitchen recipe book that matters to you. So you have all of it, but you end up with a set that's like, these are the things I make in my family. So that kind of what I actually make, that is the key. And so this process here, this epigenetic process, this pattern determines the phenotype of each cell. So it is going to cause your cell to become specialized. So the specific pattern that is actually created by the choice of what to regulate and what not to regulate is what actually determines that cell's specialization, which also determines its phenotype, or the physical appearance of that cell. So a bit of terminology. You already know all of this stuff from D1 in terms of transcription, translation. So now what we are picking up is this concept of what determines which genes are used. So we can't express everything. What we express is really important because that turns on the cell and makes it kind of specialized. So like I said, there's essentially 3 main levels of regulation that we're going to focus on. And the goal is always to regulate it when possible at the higher levels. So it's what that's meaning is if I lent you my cookbook and I was like, have a go at copying out any recipes you're interested in, the smartest thing for you would be to decide right away which ones you would never want to use and not copy them. So it'd be a waste of your time to copy things that you're not planning on using. So be smart about what you transcribe. So that's what our cells generally try to regulate as much as they can at the DNA transcription level, but there are processes that allow for regulation from there. Okay, so I'm going to erase just so I have more space. I don't think spacing is as tricky on this one, but this next section is probably going to be close to half. So kind of from what you already have through to what I'm about to do, try to leave about half left after that. Okay, so most of the time, what our cells do is in the process of becoming specialized, there's already going to be a culling. So if it is becoming a liver cell, it is never going to need the genes to be a heart cell, an eye cell, a skin cell, a hair cell. So as cells start to specialize in utero, what they want to do is go ahead and lock out all of the genes they know they're never going to use and get rid of them. So that would be like me buying a brand new recipe book and just savagely ripping out every page that had seafood because I don't eat seafood. So I'm like, no thanks, no thanks, no thanks. Don't ever need those. And so it's the removal or the discarding, but it doesn't actually remove it. So that was a bad terminology. It would more be like if I folded it over. But basically getting to the point where only the DNA that you might use is still kind of available to the cell. So let me explain that better because I don't think I explained it very well. So when we look at kind of like I said, we're going to look at this at 3 levels of regulation. And so this level of regulation is going to be at the DNA level. So one option is that we can regulate what is going to be transcribed with DNA changes. So this case, we are going to actually change the original DNA. So we are going to actually go ahead and at the original DNA point, make some changes to it. Those changes that are made to the DNA but not to the sequence, so non-sequence based changes to the DNA are called epigenetic tags. So an epigenetic tag is some sort of alteration of the structure of the DNA without changing its actual base sequences. You only have to know one type. So the one type that you need to know is DNA methylation. So DNA methylation is an epigenetic tag. It is a change to DNA, not to the base sequence, but to the DNA itself. And that's the one that you need to actually be able to talk about. And then we'll look at some kind of implications from that. So when we do DNA methylation, it can be at a couple different places. So we can actually do it gene by gene, or sometimes if there's whole sections of genes that cell is not going to use, it will actually methylate the histone. So it'll go ahead and methylate a chunk of it as opposed to one by one. So some sort of kind of turning off. So DNA methylation can do different things. So it can, in some cases, actually increase transcription. So it can kind of, anyway, it's quite confusing. The example that you need to know about is turning genes off. So it can, in other instances, actually kind of block other transcription factors. So it could block a silencer. So there's complicated ways it can work, but we're going to go through for now just the simplest basic way. So there's my promoter. Here's my gene. And so what happens is at the promoter, these little methyl groups get added. So methyl groups are CH3, and they are going to bind here at where there is a CG combination. So that's what they're able to bind to. So they're going to bind to the promoter, and they're going to put that there. And by having those bound to the promoter, the polymerases and transcription factors can no longer bind. So it basically is going to block the promoter. So now the promoter is blocked. And this gene is essentially turned off. So what is going to happen is that during embryonic development, this is going to happen, and this is going to lead to cell specialization. And cells can actually methylate up to about 90% of the genes. So as it's becoming a liver cell, it's like, I only need 10% of these, methyl. So I can methylate a lot of genes so that that certain type of cell can then become specialized. So it occurs 2 times. So it occurs during embryonic development. And when it happens in embryonic development, that is actually leading to cell specialization. But the other thing that can happen is it can happen during the lifetime as well. So it can happen during lifetime as a result of experiences. And so this is a way in which our DNA changes its pattern of expression, so not its bases, but based on our exposure to things, we can actually change our pattern. So it could be that genes that were intended to help you grow tall, but if you're in a malnourished situation, the body ends up actually methylating those genes. So as a result of kind of environmental triggers, additional methylation can happen during the lifetime, which is going to be important. Because of the fact that it happens during the lifetime, what's interesting is that monozygotic twins have the same genome, but they actually have different methylation patterns, particularly as they get older. So initially they're going to be very similar, but as they get older, their experiences are going to differ, and they're going to end up methylating different genes as they kind of adapt to their environment, as they kind of cope with their environment, and so it ends up becoming more different. So monozygotic twins are going to have same genome but different epigenomes, and again, particularly as they get older. So as we get older, we turn off and on genes differently based on our experiences. Okay, so we're gonna kind of go from this. We need to talk a little bit more about epigenesis. Epigenesis, whatever you want to call it. We'll go back to kind of other ways of regulating. But now let's talk a little bit about these epigenetic tags. So epigenetic tags should be maintained in mitosis. So in theory, once a cell specializes, it keeps those tags. So as it goes through mitosis, it holds on to the tags. And so when it actually replicates the DNA, it matches those tags. So it went in as a liver cell and it becomes another liver cell. So it still has all the genes, but it holds the tags. But in theory, when you form gametes, the gametes should remove methylation patterns for most part. So methylation patterns should actually be removed during gamete formation. And so then in theory, you start with fresh. But that is not totally the case. So there is some evidence that epigenetic patterns, so what has been turned on and off, in some cases can get passed down to offspring, which means we can genetically pass on experiences. So that concept is a big area of research right now called epigenetic inheritance. So epigenetic inheritance is the passing on of epigenetic tags. So usually it should happen that they get removed. So most but not all tags are removed in meiosis. So any of them that are not removed, so the key is in this not all. So tags that remain, so passing down methylation patterns is an example, is basically a way of inheriting experience. So when that happens, we're basically passing on consequences of experience. So the example you have to know is pretty simple, but it's actually a really, really interesting area of research into PTSD and all kinds of like trackable methylation pattern changes from Holocaust survivors through many, many, many generations. So really, really interesting area of research. The example that you need to know per your syllabus is air pollution. So what they actually found is that exposure to air pollution actually removes, I have to get them right, they actually remove methylation patterns. So if you're exposed to air pollution, it will generally remove methyl groups. And the result of removing methyl groups is more proteins, and that includes things like inflammation or so the number of proteins increase, and that tends to increase inflammation. So the air pollution is actually just a chemical thing. It's not your body intending to do it, but some of the chemicals that are in air pollution end up removing methyl groups. So then your body starts overexpressing proteins that it doesn't need to, and the consequence for the person is this kind of increase in inflammation. What they have found is that if a woman is exposed to air pollution while pregnant, then the baby is actually born with overall less methylation. So it impacts methylation in utero. And then when that baby makes gametes, it has altered methylation patterns itself, and then those can get passed on. So this is particularly problematic in pregnant women. And sorry if some of this stuff is a little bit confusing. It's actually a really complicated area that we are doing a relatively simple dive into. So for your sakes, you just need to know that one example of epigenetic inheritance is the idea that pregnant women who are exposed to air pollution tend to have babies born with decreased methylation, and as a result of their decreased methylation, they have higher inflammation, so that can lead to kind of autoimmune diseases and so forth. And then if they pass on their less methylation to their offspring, it can happen over multiple generations. So that's one thing you need to know about. The next thing that also relates to epigenetic inheritance and tags is this idea of imprinted genes. So this comes down here to this same not all. So essentially in some cases, the pattern of kind of restoring epigenetic tags differs between males and females or between the two gametes. So this is basically saying that the epigenetic tags it's methylated in one gamete but not in another. So the tags differ between sperm and egg. So essentially, they should wipe all of them, and then as the actual gamete starts to form, certain tags will be added back, and sometimes a tag is added to egg but not added to sperm, based on kind of adaptations and advantages. Sounds weird. All you need to know for this is the example. So the example you need to know about is ligers and tigions. So essentially there is a gene in lions that make offspring big. And so for male lions, they actually leave that gene on because the general idea is evolution would favor having larger offspring. For the female lions that have to give birth to these giant offspring, it's actually painful and complications arising. So they actually turn off the gene for large offspring so that the offspring are smaller, which helps them. If a male and female lion come together, one has it on, one has it off, and it comes to a medium-sized offspring. But if you mate them with a tiger, you see the difference. So if you have instead, these are going to be ligers. So ligers are usually big because a liger is a male lion that has the big gene on. So the male lion, I don't know why I can't say lion, sorry. Big gene on. And instead of then mating with a female lion who has it off, so they equal out, they mate with a tiger who doesn't have the gene at all. So the tiger doesn't hold that gene. So as a result, the only version is on. So as a result, the offspring are particularly large. The opposite is the case if you switch it. So we use different terms because of this difference. These are tigions. So this time you have a female lion. And again, she doesn't want giant babies coming out of her. So she has turned off the big gene. So the big gene for her is off. Normally she would mate with another lion and it would end up medium. But because she mates with a tiger, it doesn't have any version of that. So then as a result of the fact that this is off, they're going to end up being small. So they don't have a different version. So that's showing an example of how sometimes methylation patterns vary kind of by gender, and that is kind of intentional for the species. By turning it off in some and on in others, it can often kind of drive offspring that have the right kind of combination. So that's an example that you need to know about that. So it's kind of a whole area of epigenetic inheritance. Sounds a bit confusing. The main thing is just that you do understand this idea that we lock DNA that we don't think we're going to need. That happens early on to make our cells specialize, but it also happens as a result of environmental experiences, either intentionally or unintentionally. So sometimes our body locks DNA or unlocks it to help us. Sometimes there's just chemicals that we're exposed to that mess up our methylation patterns. And so as a result, we end up with these unique patterns over time. Two examples that you need to know of the implications of that would be this idea of the possibility of passing on experience through epigenetic inheritance, and this example where you can look at how tagging can change by gamete, and then the lions, ligers, and tigions as the example of that. Okay, so that should be at somewhere around your halfway mark for this page. So all of this is at this first level of regulation. So first thing we're going to do is go ahead and kind of mark what do we might want to use, what don't we. So I'm going to go through my cookbook and be like, never going to use that, fold it over, going to use that, stick a tag on it. Yep, good. The tags are a bit opposite in this analogy. But anyway, I'm basically eliminating. But then even once I've said, okay, 90% of the genes, never ever going to use. Here's my 10% I might use. That doesn't mean I'm going to use those 10% every day. So if I'm like, okay, I went from 1,000 recipes to 100. These are the 100 I might want to make. But every day I might then be like, okay, but I'm only going to make like 3 today. So the next level is to keep the DNA active. So don't methylate it. But then we need to still, even with the DNA that's active and available, we need to regulate when do we transcribe it versus not. So this is basically to remove DNA we're never going to use, with what remains, the next step is to regulate when we use what remains. So that is our second step in this process. And this one's going to take most of the rest of the page. Then there's like a tiny little bit just at the bottom, so leave a little bit of space, but you can use most of the rest. Okay, so the next thing we're going to do is we're going to regulate at the mRNA level. So last time I drew a DNA, so now I'm going to draw a little mRNA here, and I know it is mRNA because I have some U's on it. So next step is that we can regulate the rate of transcription. So these are genes that have remained on. And so what we are regulating is when and how much are they transcribed. So we're regulating when that occurs. The main way that is done in eukaryotes, we'll talk a little bit about prokaryotes in a minute, but the way that we do that in eukaryotes is with transcription factors. So in eukaryotes, we talked about this in D1, but RNA polymerase cannot directly bind to a promoter. It has to have a transcription factor sitting on the promoter to allow RNA polymerase to bind. So transcription can only happen when transcription factors are involved, although some transcription factors slow down the rate, some increase the rate. So we're going to do a little diagram, and then we're going to do a little table of the different types. So we're going to draw our DNA. So a lot of the times when we have these transcription factors, they actually are upstream of the gene. So this is my promoter, and my actual gene is down this way, right? Okay? So this is before we get to the promoter. There's often some extra areas that we need to know about. So we're going to also look at some enhancer sites and silencer sites. So usually far away from the promoter, we're going to have some enhancer sites. And then there's usually also some silencer sites. So those are kind of regulation sites that are away with the promoter. So then we're going to look at 3 different types of transcription factors that relate to these. So then we are going to have transcription factors that are general, that are just going to focus on binding to the actual promoter. Then we are going to have transcription factors that are accelerants. And so those are usually going to bind to the enhancer sites. And then we're going to look at some that are repressors that usually bind to the silencer sites. So I haven't labeled that. I'm going to do it as a little key just for something different. So then below here, we're going to write type of transcription factor. Then we're going to look at where does it bind and what is its impact. So that's usually going to be like, does it start it, slow it, things like that. So there's 3 types of transcription factors, and then each of them binds to a specific thing that you need to know the name of. So this green one here represents a general transcription factor. So these are ones that just bind to the promoter. So a general transcription factor is anyone that binds to the promoter. And they are going to be necessary to start or initiate transcription. So they are going to bind, and that is going to allow transcription to begin. The next option is that there's transcription factors that enhance or speed up transcription. So those are going to be called activators. So that's that little green thing there. That is an activator. If you want to put A in that. So that is my activator. It is going to bind at the enhancer sites, which are my orange. Sorry, and I did this by color. If you don't have this many colors, you might want to label it because I'm kind of marking it that way. And these are going to speed up transcription. So these are things that are going to allow it to go faster or more copies to be made. And then we have our actual repressors. And my repressor is actually going to bind at a silencer spot. And if a repressor binds at a silencer stop, it is going to slow down or stop transcription. So we have to be careful because a lot of times we say transcription factors are things that cause transcription, and that is true for general ones, but silent repressors are a type of transcription factor that actually slow down transcription. So it's really just a protein that regulates transcription in one way or another. So these are proteins that have these different roles in terms of regulating transcription. So that's the main thing that you have to know about kind of the different types of transcription factors. The next thing is kind of where do they come from? So a lot of times these are actually things that are released inside the body, so kind of an intracellular process. So one part of the body does that, so it's like an internal thing. But they can also be introduced or regulated by external factors. So transcription factors can be regulated by the environment or by kind of factors outside the cell itself. So essentially something that's going on in the environment, some sort of experience, can trigger the release of a certain transcription factor that impacts the rate of transcription. So we're going to look at the example for eukaryotes of that happening, and then we need to look at the example for prokaryotes who don't actually use transcription factors but use operons. So in a eukaryotes, the example that you would need is related to insulin and glucose. So essentially what happens is that, I'm going to leave a little bit of space. So we have our gene for insulin. But insulin is a hormone involved in glucose regulation. So what actually happens is that we don't need insulin if you're not having any glucose in your body. So you have a transcription factor, but the transcription factor is only activated when glucose is present. So glucose, when present, activates the transcription factor. So what ends up happening is, here we have our transcription factor, and in order for it to be activated, it has to have glucose bound to it. So when glucose is present, it activates the transcription factor, it comes and binds to the promoter, and insulin is made. So then that produces insulin, and it will break down the glucose. So again, we don't need insulin if we don't have any glucose. If once it's broken down all of the glucose, it will break down that glucose and it will leave and then it will get turned off again. So it's a pretty specialized way of turning on expression just when it's needed. So that happens in our cells. It also happens in prokaryotes as well. It's just that prokaryotes don't use transcription factors. And I'm gonna, sorry. A bit annoying me. I'm just going to erase that so I have enough space. In prokaryotes, they don't use transcription factors. Instead, they use something called operons. And actually, interestingly, VCU, they have to know a lot about operons. You actually do not have to know a ton about them. The key that makes it an operon is two things. First of all, something called an operator. But the other thing that makes it an operon is it actually is one promoter, many genes. So what bacteria can do is they can actually turn on groups of related genes together. So it'd be like all of these genes are related to things I make for a birthday party. So I'm going to turn them all on, and then if I don't need them anymore, I'm going to turn them all off. So it clusters genes and regulates them as a group. Eukaryotes do not do that. We regulate each gene separately. Okay, so let's look at what it looks like in an operon. So we are going to have a promoter. And then we have an extra little piece called an operator. I'm going to make this a little bigger. So this is an extra site away from the actual promoter. And then instead of it just being one gene, it would be like 3 genes. Okay, so we're going to say these are 3 genes that are related to lactose digestion. So these are going to be 3 lactase genes. So 1 promoter for all 3 of the lactase genes. So rather than using a transcription, they kind of use something called a repressor, which we said is like a transcription factor, but I don't know, they just don't call it that with eukaryotes. But they have a protein that will sit on the operator. So remember, ours is all the way downstream. Prokaryotes are a little bit simpler, so they have this option for it to sit there. When that is the case, then RNA polymerase cannot bind, and so that is going to block this. So RNA poly cannot fit, right? So the repressor sits ahead of the promoter, and as a result, the promoter is now blocked, and it can't actually make it. So if the bacteria doesn't have any lactose in it, it doesn't need to make lactase. So this would be like the normal state. So it would just sit there and it would be preventing it. In the event that it needs to break down lactose, when lactose is available, it would remove the repressor. So lactose would have a shape that's kind of compatible to the repressor. And so lactose, when present, actually removes the repressor, allowing for lactase to be made. So in the presence of lactose, that change occurs, the repressor leaves, and it's able to make the lactase. The lactase will then break down the lactose, which means it will go away, and the repressor will come back. So it's quite similar to this example. It's just multiple genes, and there's like a special operator site for it, and it's done specifically just with this one kind of protein, which is a repressor. Like I said, if you go down a rabbit hole, there's lots on lac operon, so it doesn't look like you need to know it at a very complicated level. You just need to know it's an example of the environment, which is like the presence of something. So if you're eating a lot of glucose, that changes things. So that's just another example. It's like bacteria's version of this. You don't need to know a ton of detail about the operon itself. It is actually more confusing than this. If you want to watch an Amoeba Sisters video, they talk about operons quite effectively. So you are more than welcome to read more about it. But I think you're okay just to know it as a way of regulating. Okay, so then we're going to look at our last one. And like I said, this can be pretty tiny. So I only had a little bit of space, so this is the last step. So the reason why this is simpler is because you do not want to go ahead and transcribe things that you don't intend to use. So most of the time it's regulated earlier. So the idea is we decide if we want to make something, we copy it, and then once we've copied it, we want to actually make it, versus kind of like copying things and then throwing them in the trash. But it is an option that is certainly, if things change, in the cell, we want to make sure we have options to regulate the rate of translation as well. So even though generally the preference is to regulate at a transcription level, it is important to be able to regulate it later, so that if something changes and the protein's no longer needed, it is an option to turn off translation. So we can also regulate the rate of translation. And the way that is done is basically by breaking down the mRNA at different paces. So this is going to be done by regulating mRNA degradation. So to degrade or break down mRNA. And so that's going to happen automatically because there are exonucleases in the cytoplasm that will actually just break down mRNA. So that's going to happen. And so one way of regulating that is to actually increase the number of exonucleases or to make some sort of changes to the mRNA itself, such as a shorter poly-A tail, to speed up the rate of that. So here's my mRNA. It has a GTP cap. And then remember, it has this long poly-A tail, like so. And so in order to actually break down that mRNA, you need these exonuclease enzymes. So for instance, they're going to first have to break down the poly-A tail. They're also going to have what's called a decapping enzyme. So if you want to break down one that hasn't been used, you would need a decapping enzyme. And then once those are broken down, there's others that are going to focus on breaking down the actual gene itself. So those are exonucleases. So exonucleases are enzymes that degrade mRNA in the cytoplasm. And so what that actually means is that one way to regulate the rate of transcription is to regulate this rate of degradation. So if you were wanting to get rid of an mRNA, so it's been made, but you actually don't need that protein anymore, so you thought you needed it, but then you ate it through your diet or something like that. So one option would be to increase the rate of degradation. Then that will actually lower the amount of protein. So if we want to get rid of the protein, then we can actually do that. The alternative would be to say, oh, we need more of this protein, but we don't want to have to start over with transcribing. So you can actually lower the rate of degradation, again, by basically making a really long poly-A tail, or by just blocking the enzymes that do that, then that would increase the amount of protein that you can get. So that's kind of another area where even once something's been transcribed, there's still the option for the cell to regulate its actual expression all the way to a protein. All right, that's all things D2.2. And now on to another unrelated topic, which is osmosis. Okay, welcome to D2.3. It's essentially on water movement, so it is called water potential. Although if you're SL, you don't actually have to know that term water potential. You're just looking at it from an osmosis standpoint. If you are HL, you will have to also do the water potential side of it. So we're gonna quickly define osmosis, and then we're gonna do most of this as a big chart. So this topic is fairly brief compared to some of the others, and not as challenging, which is good. And then we'll add the water potential part at the end just for our HL people. Okay, so let's start again with defining osmosis, which briefly came up when we actually were looking at cellular transport in B2, but the actual kind of real movement of water has been saved for this. So when we say osmosis, it is a term specifically for water. So it is the movement of water. And it's the movement of water through a semi-permeable membrane. And again, just to keep revising our other stuff, we know that in that semi-permeable membrane, there are aquaporins. And the aquaporins are special protein channels that help this to happen more quickly. So it's the movement of water through a semi-permeable membrane. I want to stop and add one thing. So kind of why is water so important? So the reason why we're focusing just on water movement, it is the most frequently transported substance, and that's because of the fact that it is the universal solvent, or because so many things are dissolved in it. So it readily forms solutions. So as a way of equalizing the concentration between solutions in our body, water moves to manage solution concentrations. So we're just going to remind ourselves, because it did come up again in this one, that it readily forms solutions. And I'm just going to use this terminology that's in the syllabus, which is solvation. So we're just going to add that, just so if you saw that term, you would know what that means. And I'm not going to draw the whole concept because we went into it enough in A1, but just a reminder that the reason for that is due to its polarity. So it readily forms solutions due to its polarity. So because of its polarity, it can form bonds with both high positive and negative things, or with other polar things. So it can form things with both charged and polar substances. Okay, so that is why we're focusing on water. It's through the membrane, either through the phospholipids slowly or through the aquaporins more quickly. And then the next thing we need to pick up is the direction. So normally when we talk about passive transport, we would say from high to low. But when we're talking osmosis, there's two different ways we can talk about it. We can talk in terms of the tonicity of the solution or the water potential. So I'm going to write both here, but if you're SL, you only need the tonicity. So it's the movement of water through a semi-permeable membrane from the environment with lower solute concentration. So we say from the hypotonic environment to the environment that has the higher concentration, which is the hypertonic environment. So if you are an SL student, the way you should predict water movement should be related to these words here. So hypertonic and hypotonic. So from hypotonic to hypertonic is the way that we're going to predict it. I'm going to go ahead and add under there. So it's going to keep moving from there until equilibrium is reached. To equilibrium. So essentially, it's going to keep moving until the point of equilibrium is reached. And so again, if you are actually SL, the point you need to recognize is this point called equilibrium. Underneath it, I'm just going to add our HL terminology as well. So for HL, instead of just being able to say from hypotonic to hypertonic, we would want to say from the area with higher water potential to the area with lower water potential. So you need both. So you could be asked a question in terms of tonicity, but you also would need to be able to do it from water potential as well. And then when we say equilibrium, equilibrium is going to be the place where the two environments have the same water potential. So once the water potential of both environments is the same, it will be done with movement. Okay, so that is our definition, just being clear on what it is and the direction. So what we're going to do now is make a chart of what happens to a cell if you were to bathe it in a solution. So this is looking at essentially tissue outside of like a multicellular organism where there's other things in place. And if you were to immerse it in that kind of solution, and what would happen. So we're going to do a big chart of this that will take up it's all of the rest of your page if you're SL, so you can make your chart giant. HL, you need probably a quarter of the page at the end, just in order to cover some extra HL water potential stuff. So this is going to be, we're going to name this table Predicting Cellular Impacts of bathing, and I'm just using that term because that's the term the IB is using, to emphasize that kind of idea that the cell is completely surrounded by it. Okay, so we'll set up kind of the columns and then kind of go from there. So we need to say it depends on the solution. So we'll call that tonicity of the solution. So the prediction is going to vary based on that. Then we're going to say based on that, what is the water movement? Then we're going to say what is the impact for an animal cell? Then we're going to say what is the impact to a plant cell? And note I'm using, in theory, I should say a cell lacking a cell wall and a cell with a cell wall, because it's not just animals and plants, but it's the cell wall or not that makes the distinction. And then we're going to add adaptations. So what are some things that might help it to withstand that kind of environment? So what are some things that would prevent some of the negative impacts? Okay, so that is our table, and then of course we're going to do it for hypertonic, hypotonic, and isotonic. Go. Like so. Okay, so we'll start with hypertonic solution. So it's going to start with what happens if it's in a hypertonic solution. Remember, hyper means high. So when we talk about hypertonic solutions, we are talking about a solution that has a high or a higher solute concentration. And again, I'm just going to add to this. If we were talking about it at an HL level, we are therefore talking about a solution that has low water potential. And the reason why it has low water potential is due to the number of solutes or due to its very negative solute potential. So it has a very negative solute potential, which makes the water potential low or more negative as well. Okay, so that is what we are talking about. Now let's have a look at kind of showing that. So here will be our bathing environment. Then we'll draw a random cell. This can be my cell. So my cell is going to have 1, 2, 3 solutes. My environment is going to have lots and lots and lots of solutes. Like so. As a result, again, because we go back to our rules, so water is always going to move from hypo to hyper. So my cell is comparatively hypo. So the water is going to start coming out of the cell. Sorry, it doesn't come all the way out the beaker. Just out there. Okay, so water is leaving the cell. So the movement is going to be from the hypotonic cell to the environment. So my cell is comparatively hypotonic. So again, we said here, this one has the environment has the low. So as a result of that, the actual cell has the higher water potential. So the higher water potential of the cell. So just adding those terms in there. Okay, so water is leaving the cell. So what is that going to do? Well, if the cell does not have a cell wall, it is going to shrivel like so. So the cell is going to shrivel, it is going to shrink, it is going to lose mass. So shrivels, shrinks, loses mass. We do need some terminology here. So our IB terminology for that is going to be crenation. And that's basically just going to keep happening until you reach a situation where it's lost enough water that the actual tonicity of the cell goes up. So until essentially the solute concentration equals that of the environment. So as it comes out, it's going to be actually giving solutes there. So eventually it should shrivel enough that it is now stronger inside. The outside is diluted from the water movement and it will stop. Okay, if we look at impact on a plant cell, so plant cells are a little bit different, of course, because they do have the cell wall. but what will happen is the vacuole will shrivel, and then as the vacuole shrivels, the actual cell membrane will shrivel because the vacuole is responsible for kind of keeping it inflated. And so the vacuole becomes flaccid and the cell membrane detaches. So vacuole becomes flaccid. And the cell membrane detaches. So the cell wall stays in place, but the cell membrane can actually kind of come off the cell wall. And again, we need our term for that. So if that happens and the cell membrane detaches, we would call that plasmolysis. For when that occurs. What are some adaptations? This is probably more of an ecology thing, but essentially adaptations for this is to try and basically avoid in situations where the cell runs out of water. So this would be things like water storage adaptations. So that could be things like storing water on the camel of a hump. It can also be things like closing stomata, so limiting transpiration, things like that. So anything that's in place to kind of retain water. And then in humans, another thing is thirst is an adaptation that encourages us to drink in order to dilute the bloodstream so that that doesn't happen. That adaptation one's a bit strange. It's actually more important for the next one in terms of why I'm including that. But those are some examples of things, probably going back to our ecology thing in terms of looking at even things like rats having kidney adaptations. So there's lots of adaptations in place to try and prevent the bloodstream from losing too much water. All right, I'll go ahead and do hypotonic as well down here before we erase. Okay, so next environment, it would be if it was hypotonic. And hypo means low. So a hypotonic solution is one that has a lower solute concentration. And so again, in our HL terms, if it has a lower solute concentration, it will generally have a higher water potential. And the reason it's going to have the higher water potential is because its solute potential is not going to be as high. All right, so then now let's look at what would happen. So here we have our solution. Here we have our cell. There's my purple for my cell. My cell has some solutes, but the environment has none or very few. So we'll just give it one little solute. So it could have some solutes or it could have no solutes. So in this case, the environment is now hypotonic. So it is going to cause water to go into the cell. So from the hypotonic environment into the cell. So the water is going into the cell. So again, push this down a tiny bit. Again, this had a higher water potential. The cell has the lower water potential. And that's because it has more solutes. So the higher solutes is going to cause that. Okay, so then what is the consequence going to be to our actual cell? So if we are talking about an animal cell, eventually it will expand so much that it will actually put pressure on the cell membrane. So I'm drawing two little phospholipids that can no longer touch each other. So as the water is going in, eventually it's going to rupture that. So it's going to swell, it's going to gain mass, and eventually it's going to burst. Now it may not burst, so it depends on how hypotonic the solution is. But in pure water, it would burst. So in pure water, essentially, for an animal cell, there's no way for it to end up with an equal water potential because it's just going to keep going in from the hypotonic pure water. So in pure water, it would definitely burst. If it was a hypotonic solution that didn't have as big of a concentration difference, it might just swell. If it does burst, our vocab term for that is lysis. Okay, so what about a plant cell? So for a plant cell, again, the presence of that cell membrane, the cell wall, is going to prevent it from actually bursting. So instead what's going to happen is my vacuole is going to get really full, and then the full vacuole is going to put pressure on the cell membrane against the cell wall. So it's going to exert that pressure, and that is called turgidity. So it's going to get turgid. So basically the expanding vacuole presses upon the cell membrane into the wall. Against the cell wall. And I called that turgidity. The other term that you can know for that is that this actual pressure out is called turgor pressure. When we get to HL stuff, we're going to talk more about that and actually call it pressure potential. But for an SL level, we just need to know that that pushing out is turgor pressure. So that's the turgor pressure that is resulting as a result of that. When we get to HL, we will kind of clarify, but essentially because of the turgor pressure, a plant cell could actually be in pure water and actually reach equilibrium. So water movement could stop because eventually the pressure will actually cause the water potential to be the same. This is kind of why I included the adaptations. So if you were an animal cell that was in pure water, then again, you would have this risk of bursting. So if you're a multicellular organism, we prevent that by having kidneys that will filter water out to not allow that to happen. So we have adaptations as part of our organ systems that eliminate this. But if you are a single-celled organism, you wouldn't have kidneys and bladders helping you. So instead, you would have kind of a version of a bladder, which is called a contractile vacuole. And so it really is like a one-celled version of a bladder whereby it'll hold a bunch of water and then release it. So nothing too complicated to understand about it. So here would be like my little one-celled paramecium. So this is a unicellular organism that lives in fresh water. So because again, it doesn't have a multicellular body plan that would be helping to actually get rid of water like we do, it's going to have an internal system. So it's going to have this contractile vacuole that's going to sit like pretty close to the cell membrane. Usually has these little triangles around it. And what that actually means is that when it is starting to absorb water, the water that comes into the organism goes into the contractile vacuole and then out by exocytosis. So it's just an adaptation to allow organisms to live in fresh water, even if they're only one cell big. So it basically removes water by exocytosis. And again, generally we would see that in an organism that was single-celled because they lack the body plan to be able to otherwise deal with it in different ways. So I'm just calling it unicellular, and then I'm going to also make note that it lives in fresh water. So as a result of that, it is going to need that extra little adaptation. Okay, I just ran out of space, so we will do isotonic, but it's going to be pretty tiny comparatively. So the idea is ideally the solution is similar to the cell. So a cell that was in an isotonic solution would not go through any dramatic changes. So our goal in our bodies is to try to maintain our body at an isotonic point. So that's, again, why we have kidneys and things like that. So the goal of the kidney is to try and keep our blood isotonic to our tissues, so to keep that regulated. So that's an important adaptation that we'll cover on there. So our last option is that the solution is isotonic. So isotonic solutions have equal solute concentrations. And again, if we were looking at it from a water potential standpoint, they would have the same water potential. Okay, so there is going to be no net movement of water is how we phrase it. So we just want to clarify that it's not to say there's zero water movement. So water is, when close to the cell membrane, will enter and exit outside of aquaporins. So it is a moving molecule, but for every one that moves in one direction, another would move in the other direction. So water movement would even out. So there'd be like one coming in, one coming out. Whoa, they both went out, sorry. There. Okay, so the phrasing we use for that is no net movement of water. So as a result, there is no change to the cells, which is ideal. So our cells ideally do not want to be shifting and swelling and shrinking and all of that. So there would be no change. So I'm just going to draw a red blood cell, or whatever the animal cell is, looking happy because nothing happened to it. Similarly, there will be no change to my plant cell. So he will have a cell wall, a cell membrane, and an appropriately sized vacuole. So that's plant cells feeling very happy about that, no change. So in terms of applications, like I said, when we get to it, we'll talk quite a bit about how in our bodies, our kidneys are working to maintain this point whereby our bloodstream is isotonic. So that's a really important application. So I'm just going to write the role of the kidneys. So in most animals, there's some sort of system like that. A bit more complicated in plants. They would have to regulate it more on rates of looking at things like transpiration rate and closing guard cells and things like that. The other one that's mentioned in this section is to recognize that IV fluids. So anytime fluids are given directly into the bloodstream, they need to be as isotonic as possible. So IV fluids should be isotonic. Occasionally, they are actually slightly hypotonic to actually allow for some rehydration, but they would need to not be very, very hypotonic because it would very quickly overwhelm the cell. So generally speaking, they have a little bit of sugar or salt in them in order to maintain that. Okay, so that's all things SL. HL, you need to know a little bit more. So we'll talk water potential now for HL. So the good thing is the calculations on water potential should be super easy because you do not have to calculate solute potential or pressure potential. The more important thing will be to actually understand the implications of it and how that impacts kind of what happens to cells. So let's do our actual formula. So the formula for water potential is, this is the symbol, water potential equals the sum of solute potential plus pressure potential. And then we just need to remind ourselves what those actual things mean. So if we say water potential, this is a measure of the freedom of water to move. And we already said that the higher water potential, the more likely it is to move. The other thing that we need to say is that pure water, the water potential is 0. So all other water potentials are negative. So solutions and cells are negative. Okay, and then that is some of the solute potential. So this is a solute potential, which is related to the number of solutes. So the more solutes it has, it is going to be more negative, which means it's lower, and therefore it is going to lower the water potential. So S is for solutes. Again, it has a formula, but you will not need to use the formula, so it would be given to you. And then P is pressure potential. So this is basically an indication of the turgor pressure. And turgor pressure actually makes water more likely to leave that environment because there's pressure actually pushing it outwards. So in this case, the more turgor pressure, it's going to be more positive, and therefore that is going to increase the water potential. So that is going to raise the water potential. So this is going to lower water potential. This is going to raise water potential. So one is looking at how many solutes there are. The other is looking at the turgor pressure. Generally speaking, when we talk about turgor pressure and all that, we're looking at it for plant cells because the atmosphere, only plant cells are able to exert a pressure that is greater than atmospheric pressure. So animal cells and solutions, this is not relevant for. Okay, so let's look at two applications of this. So one is related to this idea of pressure potential. And so because of pressure potential, it actually means that plant cells can actually reach equilibrium more with their surroundings. So they're more likely to actually reach equilibrium. So this is kind of an adaptation that allows plants, again, because they don't have kidneys and many other things as we do, to actually manage environments differently and actually be able to kind of reduce movement more quickly. So let's look at what we mean by that. It's kind of what we drew above, so we're just going to list it here. So if it was in a hypertonic environment, so if it is in a hypertonic environment, then we're going to recall that that hypertonic environment has low water potential. And so what's going to happen in that hypertonic environment is water is going to exit the cell. When water leaves the cell, that is actually going to do two things. So water leaving the cell is actually going to increase the solute concentration. So it's actually going to cause the solute potential to go down because there's less water. So it's going to get more negative. Solute potential is a little bit confusing because you have to kind of think about that. The other thing that's also going to happen is that when the water goes there, you're also going to decrease the pressure potential. So that's going to be less positive. And so when you add these two things together, what happens is the cell's water potential drops until it's the same as the solution. So because you have both factors, so it could happen in animal cell as well, but they don't also have the pressure drop. So because of the pressure drop more quickly, that's going to happen. And then it will basically stop when it equals the water potential of the solution. So you're going to essentially, water is going to leave the cell. It's going to cause a drop in water potential for two reasons. And so it's going to more quickly stabilize with the water potential of the environment. Even more impressive is the fact that plants can actually stay bathed, in theory, in a hypotonic environment and reach equilibrium, which animal cells could never do if it was pure water. But they could do that even if it was pure water. So let's say hypothetically, this was pure water, so water potential equals 0, which, reminder, is the highest it can be. So then as a result of the cell being in that environment, water is going to enter the cell. And again, that's going to impact two different things. So first thing that's going to happen is as water enters the cell is going to get more dilute. So we are actually going to increase the solute potential, or it's going to have more water and it's going to be less negative. So the negative solute potential is going to get less negative because of the fact that we're diluting it. But the other thing that's really important is that when this happens, we are also going to increase the pressure potential. So it's going to get more positive. So again, if you were looking at an animal cell, this would not happen. And even though you dilute it, because you can never get it down to 0 solutes, animal cells are going to end up bursting because they cannot reach equilibrium. But for the plant cell, because particularly of this, it is actually possible for a cell that still has solutes to end up with a water potential of 0. So the cell water potential rises to 0. So the cell can actually reach the same water potential as the environment. So plant cells could actually reach equilibrium in a pure water environment. Okay, so that's kind of one implication, is understanding that concept of equilibrium. The other thing we need to be able to do with this is be able to use it to understand why water moves up in plants. So we're going to do this pretty simply. I try to use just this space that's remaining. So I'm going to have down here the soil. And then in the soil, I'm going to have the roots. From the roots, I'm going to have the stem. From the stem, I'm going to have leaves. And at the top, I'm going to have the atmosphere. Like so. Okay, so the first factor, so all of this is going to need to be such that we can move water up. So first of all, we need to state that the atmosphere always has the lowest water potential because of the fact that there are a lot of solutes. So lots of solutes, little water. As a result, it is going to pull water out because water goes from high to low. So then it's going to pull water this direction because this has a higher water potential than the air. So because that is higher than the air, water is going to move from the leaves to the atmosphere via the process of transpiration. And then by losing water, that is going to increase its water potential. It's going to decrease its water potential. And so then that's going to mean that the stem is higher than the leaves. Again, because the leaves just lost water. And so then that's going to pull water in this direction and then so forth. So then we're going to see it's going to keep moving up. And then if they lose water, then that moves it in that direction. So transpiration pull is basically pulling water to the higher water potential, which is in the atmosphere. One other thing that's really important is we need help with this step here. So it's also going to be really important that the roots have so we want that these need to be lower than the soil. So my roots have to have a lower water potential than the soil in order for it to go from the soil into them. So this is what I need to happen, is I need the roots to have the lower water potential so that the water can go in from there. And the way that that is managed is by moving solutes into the roots. So the way that this is done is by actively transporting minerals. So this is done by active transport of minerals. So essentially this is just stating that we are able to move water up a plant all the way from the roots through the leaves, and that's done with really two main effects. One is transpiration, so the low water potential in the air is pulling. That's really good unless the stomata are closed. So the transpiration only happens with open stomata. So as long as my open stomata are there, that's beneficial. But if the stomata are closed for gas exchange or they're closed to try to reduce water loss, at that point it would stop. So then the other factor is here. We need to move water in from the soil. If we move water in, that would raise the water potential at the roots, which makes it higher than the stem, and it can go up that direction. So it can keep moving in both directions. So ideally, we're doing it like this. We're pushing up from the roots by increasing the solutes, and we're also pushing up from the atmosphere. So that's just explaining that upwards movement in terms of water potential. Okay, that's all things D2.2. Like I said, it's a bit of a smorgasbord of weird concepts that don't really go together, but the unifying theme is that these are things that are changing in cells in order to kind of maintain their function. All right, bye.